The DNA double helix changed the future in 1953. Francis Crick and James Watson built this chemical model revealing the structure and beauty of DNA to the world. In doing so, they established DNA as the code for life.
DNA changed really everything. And that's why that's an icon.
I mean, for me, as an icon, it's because of its legacy. It established a whole field of molecular biology and then molecular genetics, and along the way gathered several Nobel Prizes. Unlike any other chemical model I think that's ever been created, that model is personal to you and it's personal to me, and the implications for our health and the implications of the health of our relatives and our children, it's a very personal reach-through. And now it's become a media symbol, an art symbol. It's iconic in its structure.
Yes, the making of that model is in itself a wonderful demonstration of success in terms of taking a bit of scientific work done by two very young men, one of them a postdoc, the other hadn't even finished his PhD, and turning their work into something which ultimately the entire scientific world, and indeed many people beyond it, were talking about. So as a scientist, what do you find most interesting about the actual work that Crick and Watson did to make that model happen?
Well, there are two things. First of all, the actual paper that was published that everyone remembers as having the structure of DNA in it actually contained none of their own data. What they did in producing the model was effectively take 5 or 6 pieces of information that other people had put together and published over the previous 5 or 10 years, and had 2 or 3 insights of their own.
The inspirational part of Crick and Watson's work was to conceive a double helix structure, like a twisted ladder, which explained the X-ray analysis coming from Rosalind Franklin's and Maurice Wilkins' lab at King's College London.
There's been a lot of discussion about what was the role of Rosalind Franklin, because Crick and Watson both got the Nobel Prize, and so did Maurice Wilkins, who got the X-ray crystallography going, and the X-ray crystallography created the data. But Rosalind Franklin had died by the time of the Nobel Prize, but there's often been discussion that she, a woman, was somehow biased against. What do you think her role is?
From a purely technical point of view, the DNA molecules that she was working on in the lab come in different forms. And she was much more sold on looking at one form than another form. And of course, it was the one she was almost less interested in, the beta form, which was the one that excited Crick and Watson. I don't think we'll ever know, because we've effectively only really got Watson's side of the story, and he's rather dismissive of Rosalind Franklin. I mean,
and she was actually very highly regarded in her lifetime, wasn't she?
Absolutely. She published some stunning papers. And as a technician, in terms of obtaining some of the best crystals of DNA, I think she was probably the leader in the world. And of course, we see that in the fact that that data was so clean that Crick and Watson instantly recognized the significance of it. It confirmed to them it had to be a helix. Now, other people had suggested a helix, but this confirmed it to them.
This structure showed how the molecule could divide at cell division and ensure the production of 2 identical copies. It also suggested how the rungs of the double helix's ladder are made of pairs of chemical bases.
So with that geometry, you have to say, how can you put together the A's, the T's, the C's and the G's in a way that fits together? And what they did was 2 insights. The first insight was that actually it was a particular chemical form of these bases. They would only fit in the helix when A was opposite a T and G was opposite a C. And what that meant was that you had effectively a unique way of lining up across a helical molecule. And the second insight they had was that the backbone molecule actually don't run in parallel. They run the opposite way around. So it's like 2 ways of a dual carriageway. One strand goes one way and the other strand goes the other way. But of course, it's the implications of this structure of having these 2 strands that really was tantalizingly teased at the end of the very first paper, where they say it hasn't escaped our notice that this provides a mechanism for copying, which alluded to the second paper which came along only a few months later, wherein they really explained the detail of what underpinned the model. And that is the beautiful simplicity of DNA, is the fact that you can go from one molecule to two identical molecules that are copies, which of course we know in all of biology, one cell divides into two, so 1 to 2 is the perfect, it's the explanation behind.
So in the structure of it, you have the whole process of reproduction.
Absolutely. The whole simplicity of life itself is in that chemical structure.